Hello and welcome everyone. Thank you so much for joining us today. My name is Koming Aoyong and I'm with the marketing team here at GemPharmatech. I'll be your host for today's webinar, Targeted B-cell Therapies, Challenges and Preclinical Evaluation Considerations for Repurposing from Oncology to Autoimmunity. Whether you're tuning in from a biotech company, academic lab, or CRO, we are excited to have you here as we explore how preclinical models can help you investigate, predict, and overcome the challenges of repurposing oncology-focused B-cell therapies towards treating autoimmune diseases. Before we begin, just a few housekeeping items. Today's session will run for about 30 to 40 minutes, followed by a Q&A session. Please feel free to submit your questions at any time using the Q&A box, and we'll address them at the end of the presentation. A recording of this webinar will be shared with all registered attendees, so you can revisit them or share it with your team. For those of you who are not familiar with GemPharmatech, we are a global biotech company dedicated to providing solutions for biomedical research using genetically engineered mouse models. Our portfolio is one of the world's largest, with more than 25,000 strains generated to date. We are a company driven by innovation, holding more than 40 patents worldwide, supported by a diverse team of more than 1,500 experts and specialists. We have an extensive infrastructure spread across North America and China, with 7 subsidiaries and capabilities that not only allow us to accommodate more than 340,000 specific pathogen-free cages, but support more than 6,000 custom model creation projects and 1,000 preclinical studies per year. Our newly expanded site in North America is based in San Diego, offering our North American clients increased scientific capabilities that include a vivarium facility, in vivo and breeding services, along with increased access to our collection of more than 25,000 gems. Now, I am thrilled to introduce our speaker today, Dr. Juan Liang, head of the autoimmune pipeline, who leads our scientific strategy for autoimmune models and services at GemPharmatech. Dr. Liang graduated with her PhD at the Chinese University of Hong Kong and did her postdoctoral fellowship at Queen's University Belfast. She brings extensive experience in immunology, autoimmunity, and heading translational research partnerships. Today, Dr. Liang will guide us through the current state of treatments for B-cell malignancies and examine the mechanistic similarities between oncology and autoimmunity. She will further describe the challenges translating such findings in oncology research to autoimmune disease and explain some of the key considerations when planning a preclinical evaluation study. With that, I'll hand it over to Dr. Liang to kick things off. Dr. Liang, the floor is yours.
Hi, hello everyone. Thanks for the introduction and thanks for joining us for this webinar. Today we will focus on the targeted B-cell therapies, the challenges and preclinical evaluation considerations for repurposing from oncology to autoimmunities. And as introduced, there will be four parts involved in today's webinars. And first, I would like to review the current state of treatment for B-cell malignancy and drug repurposing in autoimmune disease. Well, chimeric antigen receptor T-cell therapy has revolutionized the treatment landscape for malignancy in disease like leukemia, lymphoma, and multiple myeloma. CAR-T has shown remarkable efficacy. 6 CAR-T products are approved by FDA. For leukemia, especially acute lymphoblastic leukemia, CAR-T cell targeting CD19 has achieved high complete remission rates. And in lymphoma, like diffusion large B-cell lymphoma, CAR-T therapy has led to high objective response rates and complete remission rates. For the multiple myeloma, cancer of plasma cells, it also has seen significant progress with CAR-T therapies. Anti-targeting antigens such as BCMA, CAR-T cell therapy has enabled deep remission in many patients. For the TCR engager therapies, it also has emerged as a promising approach in the treatment of malignancies. It leverages bispecific or multiple specific antibodies to bridge T cells and tumor cells, thereby activating TCR-mediated killing of cancer cells. In B-cell acute lymphoblastic leukemia, TCE targeting antigens like CD19 have demonstrated significant clinical benefits. For diffusion large B-cell lymphoma and follicular lymphoma, TCE have shown activity in relapse-refractory settings. And in multiple myeloma, TCE targeting antigens like BCMA, the B-cell maturation antigens, also have gained big attentions. Bispecific anti-BCMA CD3 antibodies have shown impressive response in heavily pretreated patients. Well, autologous CAR T therapies has been considered one of the greatest breakthrough in the treatment of B-cell malignancy. Similar to B-cell malignancy, autoantibodies-related autoimmune disease are rooted in development and functional abnormality of the B-cell compartment. And here is a following-up study published in New England Journal of Medicine, reported on 15 patients with severe SLE, idiopathic inflammatory myositis, and systemic sclerosis. Those patients received a single infusion of CD19 chimeric antigen receptor T-cell therapy. As shown here, after infusion with the CAR T-cell, all the patients with SLE had DORIS remission, and all the patients with idiopathic inflammatory myositis have an ACI-EULA major clinical response, and all the patients with systemic sclerosis had a decrease in the score on the EUSTAR activity index. And the side effect of CD19 CAR T treatment were minimal, with no high-grade cytokine release syndrome and no prolonged bone marrow toxicity effects. In addition, the instance of infection was low, and the total IgG levels was only moderately decreased. Current approaches to treat autoimmune disease typically focus on manage disease instead of offering cure. Usually, the main concept to treat autoimmune disease remains to prevent autoreactive immune cells from attacking host tissue by broad immunosuppressive agents, especially glucocorticoid and anti-inflammatory drugs. However, CAR T-cells infusion offers a novel approach by depleting B cells. Following CAR T-cell therapy, the reconstituted B cells in the patient showed a naive B cell phenotype, and the membrane B cells were dramatically reduced. The plasmablast cell were not found after B cell reconstituted, and also the SLE-associated membrane B cells were also not found after B cell reconstituted. Resetting abnormal autoimmunities in this disease through deep depletion of B cells is a potential strategy for achieving sustained drug-free remission. Well, preparing autologous CAR T cells is expensive and time-consuming. It puts patients at risk of disease progression and production failures. Autogenic CAR T therapies have the premise of lower the cost. An exciting study on allogenic CD19 targeted CAR T therapy for autoimmune disease was published last year. And in this study, a genetic engineered healthy donor-derived CD19-targeted chimeric antigen receptor T-cell treatment achieved safe deep remission in 3 autoimmune patients. So in this study, deep B-cell aplasia was maintained for almost 2 months. This CAR-T therapy is called TYU19, and the TYU19 eliminates severe skeletal muscle damage in a patient with fracture RMNM. And also the CAR-T therapy reverses extensive fibrotic damage to critical organs in 2 patients with systemic sclerosis. It should be noted that usually the conventional immunosuppressants may slow disease progression. Extensive organ fibrosis is generally regarded as irreversible. By contrast, allogeneic CD19-targeted CAR-T therapy can reverse organ fibrosis. It offers a promising treatment for autoimmune disease. Bispecific T-cell engagers targeting for CD19 were also used for autoimmune disease treatment. In this study, 6 patients with multiple drug resistance arthritis were treated with these CD19 CD3 bispecific antibodies. After treatment, the patient showed rapid decline in a clinical disease activity in all patients. It has the decreased antibodies in patient, and also the patient showed improved server vitals in Astral and PET-CT, and the patient also showing the depletion of active membrane B cells and showing an increase of naive B cells. In some patients, disease may be caused by the long-lived plasma cells that express B-cell modulation antigen BCMA, but not CD19. So some autoantibodies remain stable even after CD19 CAR-T cell therapy. This drug, the tezlistamab, has been shown to be highly effective in patients with multiple myeloma. So in this study, the authors use this bispecific antibody. So in this study, the author uses bispecific antibody targeting for CD3 and BCMA to treat 4 autoimmune disease patients. And after the drug treatment, the patient showed the autoantibody were decreased in serum, and also the reduction of membrane B cells was observed. The plasma blasts and increased in naive B cells was also observed in patients. This bispecific antibody also improved the disease activity in all 4 patients. Then we will move to the next part. We will discuss the mechanistic similarities and the difference between oncology and autoimmunities. Autoimmunity and cancer are 2 sides of the same coin. A decrease in regulatory cells like Tregs, Brix, M2 macrophages, and MDSC leads to autoimmune disease onset. However, an increase in the stem cell subsets is associated with cancer development and progression. In fact, molecules involved in immunotolerance induction are down regulation in autoimmunity, but overexpressed in cancer. So targeting such molecules requires opposite strategy. For example, PD-1 blockades are used in cancer treatment, while PD-1 agonists are used for autoimmune disease treatment, such as arthritis. But when we compare the treatment approaches for B-cell malignancy and autoimmune disease, the therapeutic strategy share similarities. Usually, depletion of the abnormal B cells represents a common strategy to achieve disease remission. For this reason, the markers for B-cell lineages are used as target in B-cell malignancy. Those targets including the CD19, CD20, CD22, BCMA, and also the plasma cell markers like CD30 and CD138. The pathogenesis of autoimmune disease is complex. Aberrant B cells contribute to disease development. Here we take lupus as example. A hallmark of lupus is the presence of hyperreactive B cells and loss of B cell tolerance. Plasma cell expansion leads to the production of autoantibodies, and the autoantibodies, the accumulated of autoantibodies are deposited in tissues such as kidney, leading to the activation of myeloid cells and complement pathways, which in turn cause T-cell distractions. So the generally used therapeutics is also targeting for the B cells, of targeting for the, and also for the lineage markers of B cells, like CD19, CD20, BCMA, and also the other markers that expression on the B cell surface. So we would like to discuss about the unique challenges in translating findings in oncology to autoimmune disease. Although the targets and treatment strategy are similar in B-cell malignancy and autoimmune disease, preclinical evaluation of drugs for autoimmune disease still have unique challenges. The first challenger is from the nature of indications. There are over 100 autoimmune diseases. That means researchers need to develop different disease models. In the cancer study, distinct cancer models can be established using different cell lines with primary readouts such as tumor volume or fluorescence intensive for blood tumors. But in autoimmune disease, each indication models has distinct PD markers and efficacy parameters. For example, if we work on the lupus disease, usually the PD markers and efficacy parameters, including autoantibodies levels in serum, the protein urine, the change in immune cell population, and the renal histopathological changes. If we work with arthritis disease, the models usually the PD markers, and efficacy parameters usually involve including clinical score, pulse thickness, immune cell population, joint histopathological change. Also, when we do the study to evaluate drugs that treat for autoimmune disease, a solid understanding of the pathogenesis is also very important for the selective disease models. It is well known that no single animal model is perfect that could resemble all the pathological mechanism observed in human patients. Instead, different models mimic distinct aspects of the pathogenesis from specific perspective. For example, we still take the lupus models as example. Some lupus models are generated by activating of the c-Gas-STING signaling pathway. So that means in this mouse model, you can evaluate the drugs that targeting for the c-Gas-STING signaling pathway. And some lupus models are generated by activation of B cells, and some models are generated by activating the PDC cells. So for the BCL-mediated models, it will be suitable for BCL targeting therapy, and for the PDC-mediated lupus models, it will be suitable for PTC targeting therapies. Another example is the asthma models. Some asthma models have the activation of eosinophil, but they are, and we also have the ASCANT asthma models. This model has a neutrophil activation. So if our drugs targeting for eosinophil, we can try the false models. If our drug targeting for neutrophils, we need to choose another models. Based on this knowledge, we need to choose the suitable models to match the mechanism of action of the tested drugs. The diversity of immunotherapy modalities represent another challenge in preclinical evaluation. For CAR cell therapy, due to MHC mismatch, CAR cells must be administered into immunodeficient mice. This is a strategy we have used in cancer studies. However, in autoimmune disease, the excessive activation of immune cells drives disease syndrome development. Consequently, models must be both immunodeficient and capable of exhibiting immune system overactivations. And when we look at when we think about the TCE therapy, since TCE plays in fact through activation of T-cell mediated targeting cell killing, so the models must contain functional T cells, and the relevant target immune cells are also need to be present in the models. If we evaluate the multiple specific antibodies, given their multiple specific targets, activation of multiple immune cells is required. And if the antibodies does not exhibit cross-reactivity with mouse proteins, the model must have humanized targets through cell infusion or genetic engineering. A similar principle can also be applied in the ADC evaluation. Then we will provide some consideration for preclinical evaluation, and we will give some examples of disease models for targeted B-cell therapies. The first model I would like to introduce is the SLE, the lupus patient-derived PBMC NCG mice. For this mouse model, we infusion the SLE patient PBMC into this immunodeficient mice, the NCG mice, to establish this lupus mouse model. After infusion of the human, the SLE patient PBMC, we can evaluate the drug efficacy. This model was developed lupus by contributed by SLE patient peripheral blood cells, like the human B cells, plasma cells, membrane B cells. All those cells contribute to the disease development. And in this model, the BD markers and efficacy parameters, including the anti-double-stranding IgG level in serum and also the total IgG level in serum. We can also have the changes of immunocell populations like B cells, plasma cells, membrane B cells. And since this model just lasts for 4 weeks, it's acute models, so there is no protein urine observed in these mice. But we can still have the IgG deposition in the kidney, and we can find the immunocell infiltration in the kidney. IgG deposition is the first step to activate the complement pathways to cause kidney dysfunction. So IgG deposition is also very parameters that we can evaluate in this model. And in this model, since we infusion the PBMC into NCG immunodeficient mice, this model is immunodeficient mice, so it's very suitable for CAR cell infusion. And in this mouse model, we have infusion the human peripheral blood mononuclear cells into the mice. The mice will have the human T cells, so it has the functional T cells for the TCE therapy. In the meanwhile, the mice have the human B cells, plasma cells, and also membrane B cells. So all the therapeutic targets are involved in these models. It could be used for the multiple specific antibody evaluation, and also ADC evaluation can also be used in this model. And here's some case studies presented here. The first case is the TC evaluation. In this figure, this column is a modeling group. After infusion of SLE patient PBMC, we could observe the increased IgG level in the serum and also the increased double-stranded DNA level in the serum. After treatment, in this study, we used 2 drugs. The first drug is a bicytogen, the CD3 CD19 bispecific antibody, and the other one is a trispecific antibody targeting for CD3 and the other 2 BCL lineage markers. All those T-cell engagers could significantly decrease the IgG level and also the double-stranding levels in serum. And also, we could observe the depletion of B cell, the CD20 positive B cells, the plasma cell, and also membrane cells in spleen. And in the endpoint, we evaluate the IgG deposition in kidney. The IgG deposition increased in the modeling group, and the TC treatment could reduce the IgG deposition in kidney. And here is a CAR therapy evaluation case we have conducted in the same models. In this study, we evaluate CAR-Treg efficacy. After 2 weeks of administration of the cell therapy, we could observe the decreased IgG level and also anti-double-stranding levels in the serum, and also the B cells, the CD20 positive and CD19 positive cells were depletion by CAR-Treg. And here is also another CAR therapy evaluation. In this study, we evaluated CAR-NK efficacy. And similar to the CAR-Treg, the CAR-NK can also decrease IgG level in serum and depletion plasma B cells. Well, here is another models I would like to introduce. It's also SLE models, and this model is spontaneous SLE models. For this model generation, we overexpression human BAFF in these mice. BAFF is a regulator of the survival of autoreactive B cells. Once overreactive of autoreactive B cells, the mice will have the increased IgG level and also the anti-double-stranding levels in the serum. So the mice spontaneously develop lupus. And then when cross this humanized BAFF mice, when the humanized CD20 when this CD20 humanized mouse model and generate this double KI mouse models. So in this mouse model, it have the human CD20 and also have this human BAFF. These mice have the high expression BAFF promote autoreactive B cell. So that's how these mice develop lupus. In this mouse model, the PD markers and efficacy parameters, including anti-double-stranding levels and also total IgG level in serum. This model is the chronic disease model, so we can observe the protein urine with increased level of UACR and also UPCR. We can also observe the changes in immunocell population like B cells, plasma cells, and this model will also develop lupus nephritis at around 25 weeks of age. This model is spontaneous SLE models and is the immunocompetent mouse model with robust overreaction of immune cells, especially B cells. So this model could evaluation of antibody detecting for BAFF and CD20. Okay, so and here is the drug efficacy studies we conducted in this model. For these studies, we evaluate the antibody rituximab, that's the monoclonal antibody detecting for CD20, to evaluate its drug efficacy in this model. After 6 days of administration of the CD20 antibodies, we could observe decreased anti-double stranding levels and IgG level and also IgM level. And also after administration of the CD20 antibodies, we also observed the decreased level of CD19 positive cells, also the plasma B cells. We also tried the antibody detecting for BAFF in this mouse model, and it showed the similar efficacy, effective efficacy results. Here is another models also derived from the patient PBL. In this mouse model, we generate the myasthenia gravis patient-derived PBL NCD mouse model. So in this mouse model, we use the MG patient-derived peripheral blood lymphocyte infusion into NCD mice, and these mice will develop some syndrome that signal to myasthenia gravis, and also the human B cells, plasma cells, membrane cells contribute to disease development in this mouse model. And the PD markers and efficacy parameters, including the anti-ACH receptor antibodies and IgG levels in serum. And also we can observe the changes in immunocell population, especially B cells. And we can observe the decrease of the full lymph group. And also there will be the increased HE score in muscles. Since this model is also the immunodeficient mice, since this model is also generated based on the immunodeficient mouse model, so this mice can also be used for CAR cell therapies evaluation. And this model also has a functional human T cells, so it could be used for the TCE therapies. And the model has the multiple specific therapeutic targets involved in the model in this mice. So it can also be used for evaluation of the model specific antibodies and also ADC evaluation. Here is the modeling data we have collected. As indicated here, after we infusion the MG patient PBL into the NCG mice, we could observe the gradually increased anti-ACH receptor antibodies levels. And also we can observe the increased IgG level. The full lymph groups were gradually decreased after infusion of the MG patients derived PBL. And also we can observe the pathological change in muscles. The mice showed increased HG score, and also we could observe the muscle fiber degeneration and necrophagy. We can observe the lymphocyte infiltration in muscle cells. Besides the other three models I have introduced, we also have generated the other models that could be used for targeted B cell therapies, like the CBSA-induced membrane nephropathy models, the full-length MOC-induced EAE model, the C-liver gland protein-induced Sjögren's syndrome models, and also the peptide-induced MG models. All these models have the B cell activation, so they will be very suitable for the B cell targeted therapies. So here is the conclusion. There are some key considerations for preclinical evaluation. So when we do the preclinical evaluation for the autoimmune disease drug efficacy, we need to know well of the pathogenesis of disease models. Just as I mentioned, there is no single models that are perfect, could resemble all the pathogenesis of patients. So maybe different models has distinct pathogenesis. So we need to figure out which kind pathogenesis, which kind signaling pathway or immune cells are involved and activated in the selected models. So we can choose the suitable models to match with our drug mechanism of action. And then we need to know the PD markers and efficacy parameters we will be observed in the models. And different indications will have different PD markers and efficacy parameters. Usually for the autoimmune disease, the generally parameters will including levels of serum antibodies, the immune cell population, the clinical score, disease index, and also different indication have the specific organ, have the specific target organs. So we will evaluate the specific organ function, and also we can look at the histopathological change of target organ. And the last point is we need to think about the modality of test article. It will help us to decide which kind of models we need. Like if we are run the study of CAR cell therapy, we may need the immunodeficient mice to infusion CAR cells. And in sometimes, if we want the immunodeficient mice also have the overreactive immune cells, we can use a immunoreconstituted mouse model. And sometimes we can also use the immune component mice with genetic engineering humanized target in the mice, and then plus some specific antigen to treat the disease syndrome. So, well, here is all the consideration I would like to share. And thanks for your attention, and I'm ready for taking the questions.
Thank you so much, Dr. Liang, for that insightful presentation. Before we proceed to the Q&A, I would like to take some time to highlight the key resources GemPharmatech offers for studying autoimmune diseases. As one of the premier companies in mouse model creation with over 25,000 strains of genetically engineered mice, we have a large selection of mouse models covering a wide range of autoimmune diseases. In addition to genetically engineered mice, we offer preclinical CRO services that include custom model creation, experimentally induced models, and humanized models of autoimmune diseases. Each of our models are well validated with a clear understanding of the underlying mechanisms, and our services are highly customizable to fit your research needs. Feel free to visit us at GemPharmatech.com or send us an email at sales@GemPharmatech.com to see how we can support you in your autoimmune disease studies. Again, thank you for your attention, and we'll now proceed to the Q&A session. So I'm going to go through the questions by the order that they arrived in. So the first question for you, Dr. Liang, is, would the models be relevant for bispecific antibody targeting strategies for autoimmune diseases?
Yes, I have included some case studies in the presentation. The models for evaluation TCE should contain functional T cells and abnormal target immune cells. As I presented in the webinar, we can use patient immunocell infusion disease models, such as lupus or MG models. We can also, and in this kind of mouse model, usually we can evaluate the the B cell population and also the IgG antibody levels in the serum. And also we can look at the IgG deposition in kidneys. And besides the immunodeficient mouse model, we can also use genetic engineering mouse model that expression human targets like the CD20 and BAF double humanized mouse models. And currently, we are working on breeding these models with CD3 humanized mice to generate triple humanized mouse expression human CD3, CD20, and BAF, as well as other combination like CD3, BCMA, and BAF. So in those kinds of disease model, because they are chronic spontaneous lupus disease model. In this mouse model, you can have the antibodies level in serum, the protein urine once increased your ACR and your PCR, and also this mice can develop a lupus nephritis. So when you evaluate your drug efficacy, you can look at those kinds of parameters.
Thank you so much.
Additionally,
Sorry, go ahead.
Okay, no
Go ahead,
problem.
go ahead.
We can move to the next question.
Oh, absolutely. Thank you so much. That was very informative. So are there any differences in how B cell depletion strategies function in oncology versus the autoimmune disease context?
Although abnormal B cells play distinct roles in disease pathogenesis in oncology and autoimmune disease, strategy for B cell depletion often target similar B cell lineage markers, such as CD20, CD19, and BCMA. From this perspective, drugs that demonstrate efficacy in oncology may also have the potential to be effective in treating autoimmune disease. This may explain why CAR T-cell therapy and TCE have shown promising efficacy data in clinical studies. However, we know that the monoclonal antibodies targeting CD20 for B-cell depletion have failed in some clinical studies. This could be attributed to incomplete depletion of T-cell resident abnormal B cells, because maybe the monoclonal antibodies have the low infiltration into the target T cells, so this may limit B cell depletion. While the CAR T cells exhibit high T cell infiltration, so it may showing more complete T cell resident B cell depletion. And also for a safety perspective, the side effect of CAR T and TC treatment appear to be minimal, while it shows no high-grade cytokine release observed. While this high-grade cytokine release were also observed in a clinical trial for cancer treatment. So I think maybe it caused by the high tumor burden. The high tumor burden may contribute to high-grade cytokine release, while the target expression levels in autoimmune disease are typically lower compared to those in tumor T cells. So I think that's the difference of the drugs used in oncology and also autoimmune disease.
Thank you so much for that. The next question we have is, what are some of the safety concerns when adapting B cell therapies for use in autoimmune diseases?
As I mentioned, the existing clinical data indicate that the CAR T and TC therapies exhibit very lower side effects in autoimmune disease treatment. But we still need to watch out for the same safety issues observed in cancer treatment, it including cytokine syndrome, infection, neurotoxicity, and off-target effects on normal T cells. And in the other side, autoimmune disease are chronic and typically less life-threatening than cancer. So it will require stricter safety control than those used in tumor treatment. So if we would like to evaluate the safety problem in the preclinical studies, we can measure cytokine release in immune cell reconstituted mouse models to evaluate the safety in those. We got a lot of experience for running this kind of studies.
That's fantastic. Thank you so much for that. We have time for a few other questions. So the next question is, what biological endpoints are considered when repurposing oncology-targeted agents for autoimmune indications?
Well, it may different from evaluation using the tumor models. In preclinical studies, if this question refers to the preclinical studies, the syndromes of several autoimmune disease models only persist for a certain period, particularly in induced models. So the endpoints should be set before the disease naturally alleviates, like before the decline of clinical scores or the decrease in antibody levels. So the setup of the endpoint relies on the experience and data collected from such models. So when we're running the preclinical studies, we really need to know very well about our disease models.
All right, thank you so much for that. We have another question coming in from the audience. It is a question kind of related to the first question that we addressed is how would we create the model of choice when we want to evaluate T-cell engagers? Like, would it be a CD3 or CD19? Would the immune reconstituted model or a gene-humanized model be more appropriate for such situations? And what would be some of the key considerations when doing that?
Yes, I think we already answered the questions. So
Yeah.
it's like if we use the immunoreconstituted mouse model, because this kind of model is acute disease mouse model. So the key parameters will be the antibody levels in serum and also the B cells population changes. And also we can look at the IgG deposition in kidney because it's the acute mouse model, there will be no kidney dysfunction, so there is no protein urine observed in this model. But if we try to use the CD3 humanized mouse model, we can use a specific antigen to induce the disease phenotype, like some TR7 agonist, and also like maybe pristin or other chemicals that will trigger immune cells response. In those kinds of disease model, usually they are chronic disease model. So in this kind of mouse model, we can get the antibody levels in serum, the B cell population change, and also sometimes we can get the lupus nephritis in some chronic mouse models. So the different models may have the different key parameters to be collected.
Yeah, absolutely. So it definitely does sound that you really understand a little bit more about the disease, and kind of use them as the key considerations in planning. So another question we have is, do we look into any antigen-specific B cells using any other in vivo or ex vivo B cell reporter cell lines assays? I'm assuming. So
into antigen-specific B cells, in vivo, ex vivo, and B cell. So I don't think we have run such studies in the autoimmune disease pipeline. I think we got some. We have run some studies in the cancer treatment. We got some. We should have some B cell line that have the specific antigens, but I'm not sure about this field.
okay, no problem. Maybe we could reach out to the member who sent us the question for further discussion. But again, so I think that's all we have for all the questions we have coming in today. So before, that really wraps up our webinar today. Again, a big thank you to you, Dr. Liang, for sharing your expertise, and thank you to all of you for attending and engaging with us. Just as a reminder, we will be sending out a full recording out shortly. for you to revisit and share with your team. If you are interested in learning more about resources for today's talk, or want to schedule a consultation, feel free to reach out to us at sales@gempharmatech.com or visit us at gempharmatech.com. We hope you found today's session valuable and that we'll have a chance to support your research in the future. So thank you again, once again, and have a good day.